Dietary carbohydrates are digested into monosaccharides and then are absorbed in the small intestine. And carbohydrates can be classified into monosaccharides, which are the monomers, oligosaccharides, which are short polymers, and these polysaccharides, which are long polymers. So the small intestine can only absorb in the monomer form, that individual form. Some polymers are digestible, and the body can break them down to form those monomers that it can absorb, and other polymers are non-digestible or fiber. Most ingested carbohydrates are in the form of starch. So starch is a really high molecular weight compound made entirely of glucose molecules, and it consists of these two polysaccharides, amylose and amylopectin. So amylose is a straight chain of glucose polymer, and it's made up of these many glucose residues combined, and they're connected by these alpha 1,4 linkages between each glucose. Amylopectin, on the other hand, is a much larger and branched glucose polymer, and it contains numerous amylose-like chains that are connected together through these alpha 1,6 branch points. Most other ingested carbohydrates are in the form of disaccharides, like these here, sucrose, which is table sugar, and it's composed of glucose and fructose monomers, lactose, which is the sugar found in milk, and it's made up of galactose and glucose monomers, and other disaccharides that are ingested include maltose and trehalose. There's also a small amount of dietary carbohydrate ingested as monosaccharides in the form of fructose and glucose, but the important point is that all of the ingested carbohydrates have to be broken down all the way to that monosaccharide form because the small intestine can only absorb monosaccharides. So starch digestion technically begins in the mouth with a salivary alpha amylase that acts on these alpha 1,4 linkages, but overall it plays only a small role because food's only in the mouth for a short time, and once that salivary alpha amylase is exposed to the acidic environment of the stomach, it becomes inactive. So although it's possible that some of that enzyme that's protected in the center of the bolus can continue carbohydrate digestion in the stomach and into the small intestine, overall, salivary alpha amylase doesn't play a large role. Starch digestion really begins in the small intestine with the pancreatic alpha amylase that's the most important. The pancreatic amylase acts at the alpha 1,4 linkages, and it yields the glucose disaccharide maltose, trisaccharide maltotriose, and short polymers containing branch points called the alpha limit dextrins. These smaller glucose polymers are then further digested into monosaccharides by the brush border enzymes, so sucrase, lactase, maltase, trehalase, and dextrinase. Then the glucose monomers and fructose and galactose can all be absorbed by the enterocytes of the small intestine. Ingested disaccharides don't require digestion by pancreatic enzymes because they're already in this disaccharide form. So the disaccharides are digested into monosaccharides by brush border enzymes alone. Specific membrane transport pathways allow the uptake of the resulting monosaccharides across the luminal membrane of the enterocyte and also mediate their transfer across the basolateral membrane into the portal circulation. The primary routes of monosaccharide absorption occur through three main membrane carrier systems. So this sodium-dependent glucose transporter 1, which absorbs glucose and galactose from the luminal membrane. This GLUT5 transporter, which absorbs fructose across the luminal membrane. And then the GLUT2 transporter, which transports all of these monomers out across the basolateral membrane and into the blood. So the sodium glucose transport protein is a transmembrane protein that mediates transfer of both sodium and glucose, or sodium and galactose, across the luminal membrane. And this process occurs through sodium-dependent secondary active transport. and it allows for that transport of both glucose and galactose. So in these cases, the energy that is stored in the sodium gradient created by the sodium-potassium ATPase on the basolateral membrane is then used to transport glucose and galactose across that luminal membrane coupled to sodium. Because this sodium glucose transporter can't transport fructose, fructose absorption occurs across the luminal membrane by facilitated diffusion by this GLUT5 transporter. Then efflux of glucose, galactose, and fructose across the basolateral membrane also occurs by facilitated diffusion through the GLUT2 transporter. Indigestible starch or fiber passes on into the colon where it's fermented by colonic bacteria to form short-chain fatty acids that can be absorbed passively by the colonic mucosal cells. So an important principle to remember here is that the apical transport of glucose and galactose is energy-driven, which helps the enterocytes absorb as much of these monosaccharides from the lumen as possible. In contrast, the absorption of fructose is via passive diffusion. So this means that fructose is absorbed only by passive diffusion and it can't be absorbed against gradient, so it's more likely to remain in the lumen. This overview shows digestion of ingested starch in the lumen primarily by that pancreatic alpha amylase into the alpha limit dextrins and maltose. These molecules can then further be broken down into monosaccharides by the brush border enzymes dextranase and maltase into the final product of glucose. And that glucose can then be absorbed by the enterocytes via this secondary active transport coupled to sodium. But we said we also ingest disaccharides in the form of lactose and sucrose, and lactose is digested by the brush border enzyme lactase to produce glucose and galactose, both of which use that same secondary active transporter, the sodium-dependent glucose transporter, while sucrose is digested by the brush border enzyme sucrase to produce glucose and fructose. Glucose uses that same pathway, while fructose uses facilitated diffusion via this GLUT5 transporter on the luminal membrane. Glucose, galactose, and fructose are all then inside the enterocyte, and they are transported out the basolateral membrane and into the blood through facilitated diffusion via the GLUT2 membrane protein.